Enzymes are proteins that act as biological catalysts. They speed up chemical reactions that take place in cells. You develop a problem statement by asking yourself what you want to know from the lab, such as at what temperature do liver enzymes work at their best. You develop a hypothesis by asking yourself, if I do this, then what would happen? How does temperature affect the rate of an enzyme catalyzed reaction? If the hydrogen peroxide is at 47 degrees, then the enzyme catalyzed reaction will function at its best. We use 4 different temperatures to be exact and do it like a good procedure should be. Specific and in order: 0 degrees, 37 degrees and 47 degrees and 20 degrees. Identify variables by asking yourself what you are changing from the control group independent and what changes from the result dependent. The dependent variable is the variable that is observed and that changes in response to the independent variable. The variable that is deliberately changed is the independent variable. Constants are variables that are kept the same throughout the experiment. Control group is the group of the experiment where the independent variable does not influence the result. Procedure: we assume you are ready to go and have all the materials. Cut several about 15 pieces of filter paper to the same size, about the size of a corn. Fill the beaker with about 50 milliliters of hydrogen peroxide. Wrap a circle of paper with the forceps and dip it in the liver puree for 5 seconds. After the 5 seconds of the puree, take the filter circle of paper out and dry it on a piece of paper for 20 seconds. After the filter paper is dry, put it inside the hydrogen peroxide with the forceps and push it down to the bottom. Make sure the timer starts timing how long it takes the paper to dry from the bottom to the top of the beaker. Record on a chart how long it takes for the filter paper to reach the top. Do 3 different trials for each temperature and then repeat these steps with the 3 other temperatures. For the temperature, put the hydrogen peroxide in a container with ice. For room temperature, just leave the hydrogen peroxide alone. For 37 and 47 degrees, put the beaker inside boiling water and measure it. Conclusion: 37 degrees is the best temperature for the enzymes to work at. We believe this because that's the temperature liver enzymes work at.